Fungi are not plants. They do not photosynthesize, but must find food like animals do. Fungi belong to a kingdom of life all their own, distinct from animals, plants, and bacteria. More than 70,000 species have been described, but there are estimated to be more than 1.5 million on Earth. All large fungi, called macrofungi, are composed of distinctive branching tissues called hyphae, and their cell walls contain chitin, not the cellulose found in plants. Mushrooms are the fruiting bodies of many macrofungi. Within their gills are millions of tiny reproductive cells, or spores, that are easily dispersed by the wind. When spores find suitable habitat, they germinate and slender hyphae emerge. Absorbing nutrients and moisture directly from their surroundings, the hyphae grow into a larger colony, called a mycelium. Huge networks of mycelia permeate soil, leaf litter, and decaying wood. Mushrooms and other macrofungi make up only part of the fungal community of the forest.
Fungi have a nucleus and membrane-bound organelles, so they are considered eukaryotic. Most fungi are heterotrophs and get their energy from other organisms. Most are unicellular, but some, like yeast, is unicellular. Fungi cannot move about. Fungi may look like plants, but they are not plants, and they are different from plants in several ways. Almost all fungi do not have chlorophyll, so they cannot practice photosynthesis. They reproduce with spores, not seeds. Many fungi have cell walls made of chitin instead of cellulose like plants. Molds have cell walls of cellulose, so they are an exception.
Reproduction in fungi is by both sexual and asexual methods. Fragmentation, fission, and budding are the vegetative means of reproduction. Asexual reproduction is by spores called conidia or sporangiospores or zoospores, whereas sexual spores are oospores, ascospores, and basidiospores. These spores are produced in specialized structures called fruiting bodies. The sexual cycle involves 3 steps as follows. 1. Plasmogamy, fusion of protoplasms between 2 motile or non-motile gametes. 2. Karyogamy, fusion of 2 nuclei. 3. Spores formation, meiosis in zygote resulting in haploid spores. Classification of the kingdom fungi. Kingdom fungi classified into the following classes based on the morphology of mycelium, mode of spore formation, and fruiting bodies. 1. Phacomycetes. 2. Ascomycetes. 3. Basidiomycetes. And 4. Deuteromycetes.
Fungi have complex life cycles consisting of asexual and sexual reproductive phases. Here, we examine the life cycle of the black bread mold Rhizopus stolonifer. The black speckles that Rhizopus produces on bread are capsules called sporangia that the fungus produces through asexual reproduction. The fungus consists of a network of filaments called hyphae that penetrate the bread and absorb nutrients. During asexual reproduction, spore-filled sporangia develop at the tops of stalk-like hyphae. The sporangia and hyphae are haploid. Sporangia break open and their haploid spores disseminate. Spores travel by air to other locations and may land on suitable food sources, such as a flaky croissant. A spore germinates and sends an elongated hypha into the croissant to absorb nutrients. As it develops, the new haploid fungus can produce more sporangia, initiating additional cycles of asexual reproduction. In addition to asexual reproduction, the fungus may initiate sexual reproduction. Although Rhizopus does not have two distinct sexes, it does have two distinct mating types, designated plus and minus. They reproduce sexually when adjacent hyphae of the two types release pheromones. The pheromones trigger the hyphae to grow side branches. Note that Rhizopus generally lacks cross walls between its nuclei, and most nuclei share a common cytoplasm. As the side branches grow, nuclei stream in. A cross wall forms near the end of each branch, isolating several haploid nuclei at the ends. The nuclei are now isolated in sexual structures called gametangia. These gametangia fuse and the nuclei inside pair up. The pairs of nuclei eventually fuse as well, and the entire multinucleate structure becomes a zygosporangium. The zygosporangium develops a thick, tough wall and can remain dormant for months. The diploid nuclei within the zygosporangium then undergo meiosis, and a sporangium sprouts. The sporangium contains the products of meiosis, haploid nuclei that are incorporated into spores. When the sporangium opens, these spores will disperse and germinate to form the next generation of haploid hyphae.